with no further ado, I'm going to introduce our fantastic lineup of speakers for this part of the session. And so I am really excited to hear about Carolyn Bertozzi's beautiful work, and she's going to tell us about therapeutic opportunities in glycoscience. Take it away, Carolyn. I don't want to take your time.
Okay, thanks, Laura. It means a lot coming from a fellow glycoscientist. And I'll just start by echoing the congratulations to Bob. Bob has been a hero of mine and a role model since my days in graduate school when I first heard him speak. And my awe has not diminished in any way since that time. So it's a great honor for me to be included in this lineup as we celebrate the career of Bob Langer. So as the title would suggest, I'm going to talk about some work we've been doing in developing cancer immune therapies that target cell surface glycosylation. And for me, the story started back when I was a first-year graduate student, learning about carbohydrate synthesis and understanding the importance of cell surface carbohydrates in human health and human disease. And even back then, there had been several decades of published observations correlating changes in the structures and patterns of cell surface glycans with cancer as a disease. And one of the most prominent observations in that space was the recognition that glycans that terminate in a sugar called sialic acid, which we then call sialoglycans, often undergo an upregulation or an overproduction in the tumor microenvironment. So whereas healthy cells will have a certain density and pattern of glycans that have this pink diamond sugar as a capping structure, if you look at cancer cells, you would often find basically a thicket, a dense forest of these sialoglycans, far more populous than in a normal healthy cell. And although the observation is now more than half a century old, it was really just in the last 10 years that the community started to understand why this might occur. And the epiphany came through the lens of an emerging understanding of the important role that the immune system plays in protecting us from cancer and eradicating cancer cells during their early stage. So the immune system comprises a family of white blood cells that are coursing around in our body. And these immune cells have the job of sampling each of our cells to determine whether they're healthy or damaged. Immune cells also are tasked with discriminating between our cells and foreign invader cells. But it's just as important that they get rid of our damaged cells to protect us from various diseases, and cancer is among them. So when things are working well, when a cell starts to transform into a cancerous state because it's had mutations to its DNA, the cell will present to the immune system molecules on the plasma membrane that engage receptors on the immune cell that deliver the signal to activate and kill that damaged cell. And so the activated cell might directly punch a hole in the cancer cell and kill it that way, or it could gobble it up, or it could send out an alarm and call for other cells of the immune system to swarm and get rid of the cancer cell. And how it kills just depends on the type of immune cell. But it's a very important function that, for the most part, protects us from cancer. Of course, every now and again, the cancer cells figure out how to avoid this recognition and killing by the immune system. And the way they do that is by engaging other receptors on the immune cell, which are inhibitory receptors. And that would be this gray receptor here. So the smart or highly evolved cancer cell will present molecules on the membrane that can engage inhibitory receptors. And when inhibitory receptors get activated, they'll quench the signaling from the activating receptors and block the ability of the immune cell to respond. And this is actually an important function of our immune system because immune cells can't make a mistake and accidentally kill a healthy cell. So healthy cells will have signatures that protect them and protect us from autoimmune disease. Cancers can evolve to present these healthy signals and take advantage of this so-called checkpoint on the immune system. So This discovery was very important for the development of a revolutionary new kind of medicine to treat cancer, which are the immune therapies. And I think the most recognizable immune therapies target inhibitory receptors on T cell, the so-called T cell checkpoint receptors, PD-1 and CTLA-4. And there are now a dozen medicines that are used clinically. They're monoclonal antibodies that either block the receptor on the T cell, which would be PD-1 or CTLA-4, or the ligand on tumor cells. And by interrupting this inhibitory interaction, that allows the immune cell to activate and respond. And this was such an important discovery in the field of immune oncology that it was recognized with the Nobel Prize in 2018 to Jim Allison and Tasuku Honjo. So the link between having an overproduction of sialic acid and this important function of the immune system in protecting us from cancer, I think really hit the radar screen of people in the field of glycobiology, because it was known that we have a family of receptors on our immune cells. They're called the sialic acid binding immunoglobulin-like lectins. And that's a bit of a mouthful, so we just abbreviate them the Siglecs. And these are receptors that bind sialoglycans and can lead to the suppression of immune cells. So in humans, we have 14 members of this Siglec family. All of them are transmembrane associated proteins, where this yellow line represents the immune cell membrane. And outside of the cell, they have domains that allow them to bind to sialoglycans, where the sialic acid residue is a very important part of their binding epitope. But what was so intriguing to us and to others in this field was the fact that inside the cell, facing the cytosol of the immune cell, many of these Siglec receptors have domains that immunologists ascribe these acronyms, the so-called ITIM or ITSM domain. And these domains are the exact same domains on the T cell checkpoint receptor, PD-1, that allow it to signal to the immune cell to go quiet. So this common signaling machinery prompted us to think that maybe the Siglec receptors were putting immune cells to sleep in response to binding sialoglycans on the cancer cells. And if so, it would make sense, because a smart cancer that evolved the ability to overproduce sialoglycans would have a selective advantage in avoiding immune cell recognition. So just as the T cell has a checkpoint receptor PD-1 that binds a ligand on the cancer cell, and we block this now with our immune therapies, we wondered whether Siglecs on immune cells could be binding sialoglycans on the cancer cell, and that could contribute to immune suppression, in which case blocking this interaction could be of therapeutic benefit. So all of this work kind of unfolded between around the year 2000 and 2010. So there was a decade during which our group and many other groups published showing that various Siglecs on various immune cells, including T cells, macrophages, NK cells, that all of these immune cells can be modulated by the sialoglycans on the cancer cells. And if you're interested in a perspective on what's happened in this field, I thought CNE News did a really nice job with this article in 2019, labeling Glycoscience as a fuel for a new breed of cancer immune therapies. So in recent years, we've been focusing on how would you make a therapeutic molecule that broke up the interaction between the Siglecs and their sialoglycans. So therapeutic strategies is now the focus of our lab. Now, on the one hand, the immune cell has a Siglec. You could imagine making a therapeutic monoclonal antibody that binds the Siglec and blocks it, so it can no longer engage with those sialoglycans on a cancer cell. That's exactly how Merck's blockbuster drug Keytruda works. That's an antibody that blocks PD-1, the receptor on the T cell, so that it doesn't engage with the ligand on cancers. The problem is that there are 14 members of the Siglec family, and based on our biochemical analysis, at least 5 or 6 of them could contribute to immune cell suppression in the environment of the tumor. So you might have to block multiple members of the Siglec family, and you'd have to know which ones are the most important for a particular patient's cancer. So instead, we started by thinking about the other side of the equation, which are the ligands on the cancer cell that engage the Siglecs. And there's a lot we don't know about the actual glycoproteins or glycolipids that are involved in Siglec binding in patient tumors, but we do know for sure that all Siglecs require a sialic acid residue as part of their macromolecular ligand. So we had the idea of developing what my students would call a molecular lawnmower that we could target to the cancer cell and which would degrade the sialicides along the surface of the cancer cell and just mow them right off. And if we could do that to the cancer cell, we would deprive it of its ligands for any and all Siglecs, regardless of the Siglec or the immune cell that it populates. So how do you target a lawnmower to a cancer cell? Well, many cancer cells overexpress protein antigens that are already targets for therapeutic monoclonal antibodies. So we figured maybe we could attach an enzyme to a tumor targeting antibody where the enzyme was a sialidase. And sialdases will catalyze the hydrolysis of the linkage between the sialic acid sugar and the sugars that lie beneath it. So we started this project in the hands of a talented postdoctoral fellow named Han Shao, who happened to do his PhD work in Pete Schultz's lab, who's one of our speakers later today. He's now an assistant professor at Rice University. And after his first generation pass at making an antibody sialidase conjugate, he then trained Melissa Gray, who's a fifth-year grad student in the lab. And she built this beautiful molecule that is a fusion between the HER2 binding antibody trastuzumab, which is already a clinically approved treatment for HER2 positive breast cancers, and a sialidase from a bacterium called Salmonella. And I won't go through the details. This is all published work, but she was able to fuse the enzyme to the antibody using two bioorthogonal chemistries. So this was a chemical fusion between two proteins. And after confirming that this molecule would selectively strip the sialic acids off of HER2 positive cancer cells without touching cells that lack the tumor antigen HER2, we joined forces with Heinz Laubli and his grad student Michael Stanzak at University Hospital in Basel and tested this trastuzumab sialidase conjugate in a HER2 positive breast cancer model in the mouse. So this is a breast cancer model in which treatment with the antibody on its own is known to be ineffective. It's resistant to trastuzumab. And you can see it in this graph. This is the volume of tumors as they grow as a function of time. And mice that are treated with the antibody alone, which is the blue curve, those tumors grow as if the mice had just been treated with a vehicle, which is phosphate buffered saline. But when we treated those mice with two different doses of the antibody fused to a sialidase, now you can see the tumors have a much harder time growing. And what Melissa showed is that's because immune cells are active in the tumor environment, keeping those tumors at bay. And I should point out, just this morning, we had another paper post as a preprint on bioRxiv showing that combination of the trastuzumab sialidase conjugate with the blocker of the T cell checkpoint PD-1 can cure these mice and basically make them resistant to engraftment with future tumors. So we're very excited about this, so much so that we formed a company called Palion Pharmaceuticals, which has now generated a human clinical candidate in which they took the antibody that we had chemically fused to a sialidase, and rather they made a genetic fusion with a human sialidase that looks like this. So it's very much the same size as a normal monoclonal antibody. And we're on track to get that molecule into cancer patients probably by the end of this year or early in 2022. But meanwhile, we thought we could do a better job at more refined treatments if we could understand more about the actual ligands that support Siglec binding in vivo. And could we make therapeutics that were more specific for a subset of Siglecs instead of stripping the sialic acids off, which would affect all of the Siglecs? So toward this end, my postdoc, Simon Wisnowski, took a CRISPR screening strategy to identify the genes that were important for the biosynthesis of the ligands for individual Siglecs. to get more insight into what constitutes their ligands in the context of cancer cells. And he did this by starting with a chronic myelogenous leukemia cell line called the K562 cell, which was transduced with a library of guide RNAs that knocked down each individual gene in the genome in each individual cell. And then he sorted out from this population of genome-wide knockdowns those cells that had lost their ability to bind individual Siglecs. And he could do this by flow cytometry sorting using soluble recombinant forms of the Siglec receptors fused to an Fc domain of an antibody and carrying a fluorescent probe. And so I'll just cut to the chase that what we learned from this screen, focusing on initially the human Siglec 7, is that Siglec 7 has a very specific macromolecule ligand on those K562 cells. And it's an interesting protein called CD43, which is characterized as a mucin. A mucin is a glycoprotein that has long stretches of serine and threonine residues that are modified with O-linked glycans, clustered together so closely that the polypeptide backbone is forced into a rigid extended conformation, and this glycoprotein stands like a giant redwood tree on the surface of the cancer cell. And the CD43 polypeptide was modified with a particular pattern of sialic acid-rich glycans. In fact, we found that Siglec 7 binds to a specific cluster of residues in which serines and threonines right next to each other are modified with a carbohydrate structure known to the glyco world as the disialyl T epitope. There's a lot of sialic acid packed into a very short region here, and this turns out to be critical for the high affinity binding of Siglec 7. So a collection of grad students in my lab wondered whether we could have a more refined therapeutic that would only take off these kinds of mucins like CD43 and leave the rest of the sialic acids that were irrelevant for Siglec 7 binding intact on the surface of a cell. So they envisioned, rather than a molecular lawnmower per se, a molecular chainsaw that might come along and cut those mucins off of the cell and deprive them selectively of their ability to engage Siglec 7. And I should point out that this was a project area that started with former grad student Kayvon Pedron, who's now a group leader at HHMI's laboratory at Genelia Farm. And it's now in the very capable hands of grad student Judy Schoen, who, by the way, was an undergrad at Caltech and worked in David Terrell's lab. So thank you very much for Judy Schoen. A brilliant chemist who works closely with another Caltech graduate named Gabby Tender, who worked in Dennis Doherty's lab, and a relatively new grad student, Angel Kuo, who was a Wellesley undergrad who worked for research in a lab at MIT. And in fact, it was Christopher Love's lab, who's one of Bob Langer's colleagues. So what is the chainsaw? Well, turns out there are proteases produced by the microbes that live in our gut that will selectively cut the polypeptide backbone in mucins, these densely O-glycosylated proteins. They're called mucinases for short, and we've spent some years characterizing mucinases from different bacteria. Usually the bacteria would use them to move around in the gut and cut the mucins that make up the mucus layer. But we found that they will also cut in different glycopeptide regions, they'll cut mucins that are on the surface of cancer cells. We took a particular interest in a mucinase called STCE from E. coli, we call that sticky for short, which cleaves a motif, glycosylated threonines in a little cluster, that is observed in a number of mucins, importantly in CD43. And this is just a Western blot showing a variety of mucins. When treated with sticky, will get chopped into discrete little bands. And so we wanted to engineer a sticky that would only cut the mucins on cells that we targeted it to, and not any old cell. So to do this, we actually did a protein engineering project where we successively lopped various domains off of sticky and also made a mutation in the active site to arrive at a mutant that we call ddSic W366A. And it's basically a compromised version of the mucinase that is inactive on cells unless you park it right on the surface of the cell whose mucins you want to cut. And so to get that parking functionality, we fused our relatively dead sticky enzyme to a nanobody that binds HER2. So this is now a reagent that will park on the surface of HER2 positive cancer cells and only take the mucins off of those cells. And here's some graphs showing how we can cut CD43 off of K562 cells that are either lacking HER2 in the top graph or expressing HER2 in the bottom graph. And the important curve to look at is the red curve. This is our compromised sticky with the nanobody on it, which will not remove CD43 from regular K562 cells unless the doses are very high in the hundreds of nanomolar to micromolar range. But when HER2 is expressed on these cells, now that nanobody sticky fusion is very active at cutting off CD43, just as active as the wild type enzyme that hadn't been damaged. So this is a reagent that we can now use to take off mucins selectively from tumor antigen expressing cells to see if we can't potentiate the ability of immune cells to recognize and kill them. And this is hot off the press data that Judy sent me, showing that a mixed population of HER2 positive, which are red, and HER2 negative, which are blue cells, mixed together with immune cells called NK cells and our dead sticky nanobody conjugate, can lead to selective killing of the HER2 positive red cells, preferentially over the HER2 negative blue cells. So that gives you a flavor of where we're going with this project. But just to summarize, I started by explaining that over the last decade, the community recognizes that tumor sialoglycans are engaging Siglecs on immune cells, and that this drives immune suppression and allows tumors to thrive. And we can undo that by targeting sialoglycans on the tumor cell surface with antibody sialidase conjugates. Hopefully this will lead to better treatments for patients. And then in recent work, we identified a mucin, CD43, carrying disialt epitopes as a discrete ligand for Siglec 7 on certain tumors. And we're hoping that we can develop these targeted mucinases as more targeted therapeutics in this axis of Siglec biology. And with that, I'll conclude. And once again, thank everyone who invited me to the symposium and to share in the celebration with Bob Langer. Congratulations on your prize.
Thank you.
Thanks, Carolyn. That was terrific. And I'm gonna just remind people, if you do have questions, to please put them in the chat. But let me go ahead and ask you. I think that's I love the mucinase work that you're doing, and that's a really clever strategy. I'm just curious, like, what kind of, and this is very translational, but I know that's where you're headed. What kind of cancers do you think would be best treated with this approach? And do you have to worry about sort of, I'll just start there.
Okay, yeah. I think the most logical cancers that would benefit from this kind of a therapeutic would be highly mucinous cancers. Yeah. So there are mucins. CD43 was a hook for us to get into this mucinase business, but it's one of several cancer-associated mucins which have been shown to be drivers of immune suppression. So breast cancers that overexpress MUC1 would be another category of tumors. Leukemias, many leukemias overexpress CD43. It's been a negative prognostic indicator for 2 decades. And then, of course, MUC16 is overproduced by a majority of ovarian cancers. So those would be the 3 indications where you might want to target a mucinase. It would be easier to target a mucinase to breast cancers if they were HER2 positive, and to leukemias, for example, if they were CD20 positive. Ovarian cancers have a number of markers you could envision targeting, yeah.
And then the mutation you made, is that kind of a conserved throughout the mucinases, or are you going to have to think about mechanism for each enzyme?
Yeah, I think each enzyme will be its own beast. So sticky has the benefit of being very broad spectrum in terms of the glycan modifications that it will tolerate when it cuts. It has the downside of being extremely efficient on its own, which is why we had to cut off its arms and legs to render it sort of helpless. Other mucinases we've looked at are less inherently active, which makes them a little less dangerous, but they tend to then be a little pickier about the glycosylation structures, where they won't act, for example, unless you first take off the sialic acids, right? So it's sort of, I think a dead sticky is probably better than a live picky mucinase.
That's good. Pengwu has a question. He wants to know, is disialylated CD43 also found on tumor infiltrating T cells?
Hi, Peng. And yes, it is. In fact, CD43 in the disialyl glycoform is known to be upregulated on T cells when they're activated. And no one has really had much of a mechanistic insight into this, but it's entirely possible that that glycoform is part of the resolution phase of inflammation, where it's basically helping an activated T cell figure out how to quiet down, right? Originally, in the early days, we thought that could occur by cis ligation with Siglec 7 on the T cell, Siglec 7 also being known to come up during T cell activation. We looked at that, and in fact, we see no evidence of that cis interaction. So it might be that CD43 and the disialyl T epitope on the activated T cell is actually involved in CD28 binding. And Jim Paulson, of course, is building that story right now. He had a bioRxiv preprint a few weeks ago on that. So we're watching that really, really closely. Thank you.
And then there's another question here that says, can you exploit the recognition of sialic acid itself to couple the mucinases?
Oh, that's a really interesting idea. Possibly. There's two things that would be interesting. If you could couple a sialidase and a mucinase together, you get a pretty interesting punch on the cell. And I don't know what the consequences of that would be, but it would be something you could one could certainly look at that. Another interesting question is how much precision can we get? So right now we park these enzymes on a cell and then they just mow everything, it's like a weed whacker, right? Everything on the cell surface takes a whack. It would be even cooler, I think, if you could tie the mucinase directly to an individual molecule and target a molecule to be cleaved, like CD43, without cleaving any other innocent bystander mucin that happened to be in the vicinity. So that kind of unimolecular precision is something we're going to go for.
Yeah, that is, I think that is something that I was curious about, like how many other mucin-like molecules are on the cells, because I think a lot about mucins that get deposited, but are there a lot of mucins we want around on the cells?
Possibly, yes. There's definitely, most cells express many either mucins or proteins that have a domain that's like a mucin-like domain, and it's a susceptible domain. On a cancer cell, you tend to be not so empathetic with what you cut, but you certainly wouldn't want, indiscriminate cutting of mucins from other cell types that you want to spare. So yeah, I would worry about having too hot of a mucinase.
Well, I know you guys have your lab has identified other mucin-like domains that were initially cryptic. So
that's right. Yeah, mucin domains are lurking here and there. Right. So one would like to protect the good guys. Yeah.
Yeah. So that I think your lab is basically uniquely set up to identify to distinguish the good and the bad.
Yeah. We'll see. Yeah, right. We're going to try and figure that out.
Well, thanks so much for that really excellent talk. It's really exciting directions for both the glycobiology field and the cancer field.